Aloha everyone, welcome to the start of our lectures on chapter number 9. So we are going to be moving into muscle and muscle tissue. So we've covered bones, now we're moving into our muscles. So we're going to be trying to make the connections with the bones that we've talked about in chapters 6 and 7. So we're going to be learning the anatomy and the physiology of muscles in chapter 9, how they move and how they work. So today is going to be a little bit of an overview of these muscles. We're going to go deep into the micro part of muscles and learn how it works down to the cellular level, because you need to really understand that in order to understand how our muscles are going to contract and relax, and then how the movements of our sarcomeres work. So with this, you're going to recognize some of the terms that we actually learned clear back at the start of the semester when we talked about the parts of the cell, because we're going to see that muscle cells have those similar parts of the cell, but they have particular names that let us know that they're parts of muscle cells, which we'll learn today. So that's why learning kind of that review of basic biology at the start of the semester was so important, because it's important for us to know all the parts of the cell as we study all the parts of the body, even for muscle tissue, because that muscle tissue is going to be made up of cells. So this lecture is going to be chapter 9A, looking at muscle and muscle tissue. You can see that we have a lot of learning outcomes for today. To kind of break it down, some of the things that we're going to be learning is we'll be learning about our skeletal, cardiac, and smooth muscle tissue. You may remember that we actually briefly talked about those at the end of Unit 1. So we'll be touching on some of the differences between those three types of muscle. We're going to be learning mostly about skeletal muscle in this lecture, and we'll briefly be touching on cardiac and smooth muscle in the next lecture. We're going to be comparing and contrasting the important functions of muscle tissue. We're going to look at how the structure of that muscle tissue is organized. We'll talk about the connective tissue that's going to connect the different muscles. And then we'll look at the different skeletal muscle fibers. And then at the end of today's lecture, we're going to kind of start to get to talking about the contraction and relaxation process of the muscles actually moving. And then we'll talk more about that in our next lecture as well. So looking at an overview of our muscle tissue. What is a muscle tissue? Well, muscle tissue is going to make up nearly half of our entire body mass. And the muscle tissue uses up a lot of energy, or a lot of ATP, that adenosine triphosphate that we talked about back in Unit 1. So we learned how ATP is generated, and it's going to be converted into mechanical energy from chemical energy. So we start with that chemical energy, convert it into mechanical energy so we can do that work. We really didn't completely go over cellular respiration in this course. I'm sure in a basic biology course you did. But as a reminder, cellular respiration is what that mitochondria undergoes to generate energy for the cell. And it does so, that energy is in the form of adenosine triphosphate, which is shortened to ATP. So ATP, again, is what our body uses to do any chemical processes in the cells. And this is generated a lot by our mitochondria through the different processes of glycolysis, the Krebs cycle, and the electron transport chain. So those three different portions of aerobic cellular respiration. I'm not going to, spend our time today going into cellular respiration, but there is a really good bioflix in Pearson that you can look at to learn more about cellular respiration, or if you just want to kind of review over it. It is a basic biology concept, so not something we went over in this course. But if you'd like to check that out, it's good. Everything on Pearson is a really good resource for you. So this does a review of cellular respiration. Back to talking about muscles. When we're looking at different muscles, we're going to be looking at the different types of muscles. We're going to be looking at the characteristics of those specific muscles and then the function of those muscles, because they're going to differ depending on where they are in our bodies. Before we talk really in depth about the different types of muscles, there are a few terminologies that you should be familiar with. So anytime you see these prefixes right here, myo, mice, and sarco, those are going to mean muscle. So I put an example here for you, the sarcoplasm. This is simply going to be the muscle cell cytoplasm, because again, sarco means muscle, right? So this is the cytoplasm that is in a muscle cell. So you're going to see that come up as we talk about the different portions of skeletal muscle as we move into this lecture. As a reminder, there are 3 different types of muscle tissue. We have our skeletal, cardiac, and smooth. And we briefly talked about these 3 types of muscle tissue when we looked at chapter 4, which was all about tissues back in unit 1. But again, the difference is that with skeletal and smooth muscle cells, these are going to be elongated, and they're actually referred to as muscle fibers. So the cells themselves are called muscle fibers. And that structure of the skeletal and the smooth muscle has a completely different function as compared to cardiac muscle. So we're going to look in the next couple slides a little more in depth at these 3 different types of muscle tissue. So this slide here is showing you a comparison of the 3 different muscle types. So skeletal muscle, which you see first here, is packaged into skeletal muscles. So skeletal muscle tissue is packaged into these skeletal muscles. And the skeletal muscles are going to be organs that are actually attached to the bones in the skin. So skeletal muscle fibers are going to be the longest of all of our muscle tissue, and they have these striations that you see. Those striations are just stripes going up and down the muscle fibers. Skeletal muscle is also called voluntary muscle, and this is because it can be consciously controlled, right? I can tell my arm to move up, right? I can tell my knee to lift up in the air. That is under voluntary conscious control. Skeletal muscle is going to contract very rapidly. It tires easily due to these rapid contractions. And some of the key words I'd like you to remember for the skeletal muscle here is skeletal, striated, and voluntary. So again, skeletal muscle is going to attach to the bones or the skin, and you can see these striations here, very long cells, the longest of all of our muscle tissues, and you can see it's also multinucleated, so multiple nuclei per each cell. And again, the cell is also known as that muscle fiber. Then we have our our cardiac muscle is next here. So cardiac muscle is only going to be found in the heart, which makes sense, right? Cardiac means heart, and it makes up the bulk of the heart walls. We see that cardiac muscle is also striated. You can see these stripes here. So just like skeletal muscle, it is striated. But unlike skeletal muscle, cardiac muscle is involuntary. So this means it cannot be consciously controlled. It contracts at a steady rate due to our heart's own pacemaker, but we can see an interaction with the nervous system as well. So the nervous system can actually increase our heart rate. So say you get really shocked or really scared by something, then of course your nerve muscles will recognize that and they see it as unfamiliar, and that's when your heart starts to pump a lot faster, and that's how we see this interaction between our nervous system and our cardio system. So some of the key words here for our cardiac muscle are going to be cardiac, striated, and involuntary. Then we have our smooth muscle over here. So the smooth muscle tissue is found in the walls of hollow organs. Examples would be things like our stomach, our urinary bladder, and our airways. Smooth muscle is not striated, right? You don't see any of those stripes here. There are no striations. And it, like cardiac muscle, is going to be involuntary. So that means we cannot control it consciously. So, sorry about that. Things like, think about your food moving through your stomach to your small and large intestines and out of your rectum. This is going to be an unconscious effort, right? We don't sit there and tell our food to be digested. Our muscles are just pushing that along through the intestines all the way out without you even thinking. So some of the key words for smooth muscle are going to be visceral, which visceral means organs, right? Because this is going to line the walls of the hollow organs. And other keywords are non-striated and involuntary. So only skeletal and smooth muscle fibers, as a reminder, are going to be elongated and they're referred to as muscle fibers. For a heart, for that cardiac muscle, if the muscles were to be so much more elongated, it'd be a lot harder for our heart muscle to pump the blood through our body. So we need the length of the cardiac muscle cells to be much more limited. It's not as elongated as these other muscles, the smooth and the skeletal and smooth, because we need a much faster contraction with our cardiac muscle. So that was a brief review of the 3 different types of muscle. And as I mentioned, today we're going to be mostly focusing on skeletal muscle. Skeletal muscle is packaged into, skeletal muscle tissue is packaged into skeletal muscles. So again, these are organs that are attached to the bones and skin. So for example, your Achilles tendon, that's going to attach to your calcaneus, and that is going to be the tendon that actually attaches your calf muscle directly to the bone to be able to move that. So the skeletal muscle fibers, again, are the longest of all of our muscle, and as a reminder, they're going to have striations, or those stripes, right? They're going to contract very rapidly, they're going to tire easily, and they're very powerful. So this figure over here on the bottom is showing you the different layers. So we have the skin right here as the most superficial layer. We talked about that back in chapter 5. Then we have that subcutaneous layer in yellow. Then we have the muscular aponeurosis, aponeurotic layer right here. And that is actually how a lot of our muscles are going to attach indirectly to the bone. It's through this aponeurosis. There's another layer beneath that. So this 4th layer here is going to have your ligaments and spaces to help kind of secure the muscles. And then just deep to that, number 5 here is going to be the periosteum and that deep fascia. So that periosteum is going to be the bone, right? And then that deep fascia is going to be in between the muscles and the bone. So just to kind of make sure that you have a good grasp on what exactly is classified as a skeletal muscle, we have this kind of review slide here. The question is asking you what muscles are skeletal muscles. Your different choices are erector pili, your tongue, the muscle in your gallbladder, and the muscles that control movement of the eyeball. So for A, the erector pili, do you think that is smooth, cardiac, or skeletal muscle? So when you get cold, do you tell the hair on your skin to stand up? No, right? So the erector pili are actually smooth muscles. They're going to attach to the base of the hair follicle and the dermal tissue. So when it's cold, the muscles are going to contract to generate heat, and that hair is standing up straight on the skin. So this is a smooth muscle. What about your tongue? Can you, if I tell you to, can you just stick your tongue out? Yes, right? So the tongue is a type of skeletal muscle. It is under conscious control. What about the muscles in your gallbladder? We can't control those, right? So this is smooth muscle. And then D, the muscles that control the movement of your eyeballs. Can you move your eyes side to side? Can you roll your eyes? Yes, right? So it's B and D that are going to be your skeletal, skeletal muscles. So next, we're going to move on to looking first at the characteristics of our muscle tissue, and then on the next slide, we're going to talk about the functions of our muscle tissue. So what are the things, in other words, that make muscles different from all other tissues? First of all, in terms of the four main characteristics that all muscles share, the first one is excitability. In other words, the responsiveness. So the ability to receive and to respond to stimuli. So our muscles have the ability to conduct electricity. And this electricity in the body is the movement of ions, mainly sodium, that's going to allow the muscle to respond to stimulus. That's why, especially when you're working out, you're going to be using a lot of this electricity and you're conducting a lot of electricity in the body because of that intense movement. So a lot of the time you're going to be losing things like your electrolytes, your sodium, right? And that's why you're always told after, say, you have a game or you have an intense workout, you're told to drink things like Gatorade or coconut water because they have a lot of electrolytes to supply things like sodium back to the body. The second key characteristic is the contractility of muscle. So this is the ability to shorten forcibly when stimulated. So a muscle can shorten, and as it shortens, it also thickens. So that is a huge difference right there between muscle cells and other cells of the body. Thirdly, we have extensibility. This is that ability to be stretched. So this is the ability to stretch without breaking. And then we have elasticity, which is that ability to, after being stretched, to be able to recoil to the resting length. So with that elasticity, we see that in most cases, muscles can be stretched and then spring back. And that's really important because other tissues do not have that ability. But because our muscles do have that ability, we're able to grow our muscle, right? We're able to build muscle, and we're also able to make muscles smaller without actually damaging the muscle tissue. So these are four main characteristics shared by all muscle tissue: excitability, contractility, extensibility, and elasticity. Next, when we look at muscle function, we see that there are four important functions of muscle. First of all, our muscle is going to produce movement, right? That's the one that you think of instantly. So muscle is responsible for all locomotion and manipulation. So even all the simple things that we don't even think about, our muscles are going to be involved, right? Every step you take, right? Every time your heart's beating. Every time you're breathing in and out, right? That takes muscles to do that. So everything, walking, digesting, pumping your blood, all of that is going to take muscle. Secondly, we need our muscle to maintain our posture and our body position. So just think of our axial skeleton structure. So again, that's going to be the skull, the spine, and the ribs. So all of that by itself is not going to be able to hold itself up, right? That's why our muscles are there in between all those bones to help us maintain our posture. Thirdly, muscle is really important for stabilizing joints. And you may remember in chapter 8 when we talked about joints, I had that slide that told you 3 different ways that we can increase the stability of our joints. And the most important one on that slide, if you remember, is by increasing your muscle tonicity, right? So a lot of joints are very fragile. So these muscles are going to surround our joints in order to stabilize them and protect them. And again, that muscle tonicity is the most important factor to increase joint stability, as we talked about back in chapter 8. The fourth function here then is to generate heat as they contract. So when we contract our muscles, they're going to generate heat, and that keeps us warm. So think of anytime you're cold, what's kind of the first thing you instantaneously do? You shiver, right? Because you're cold. And we shiver in order to increase our body heat, and this helps us to maintain homeostasis. So again, these four main functions here of muscles: producing movement, maintaining posture and body position, stabilizing our joints, and generating heat. In terms of our muscles, they are equipped with great nerve and blood supply. So muscles are vascular, meaning they have blood vessels, and they are innervated, meaning they have nerves. Innervated means we have consciously controlled skeletal muscle that has nerves supplying every fiber to control the activity. Vascular means that the contracting muscle fibers are going to require huge amounts of oxygen and nutrients. We also need the waste products to be removed very quickly. So each muscle is going to receive a nerve. So in yellow here, again, all these diagrams, if you see these yellow lines, those are going to be nerves. The arteries are usually always red. You see that here. And in blue, that's going to be our veins. So this is right now in this picture, we're looking at the femur, right? So this is the femoral vein right here. And you have your nerve, artery, and vein, right? So consciously controlled skeletal muscle has nerves supplying every fiber to control activity. And that's why we need a lot of nerves, right? Because we need to be able to control certain muscles just in order to do our daily functions. Contracting muscle fibers, as I mentioned, are also going to require huge amounts of oxygen and nutrients. We're also going to generate waste products that we need removed very quickly. So let's take an example. Say I'm working out. I'm using a lot of energy, right? I'm using up that ATP. I'm using up a lot of oxygen. Maybe I'm going out on a long run, right? So that oxygen needs to come in, and all those nutrients need to come in to supply my body, and that's going to be triggered in my system, right? I'm going to be supplying that. It's just part of the process as you're working out. But when my body is depleted of oxygen, my muscles become depleted of oxygen, right? And my muscle cells, if they don't have oxygen, they're not going to be able to do aerobic respiration, right? My muscle cells are going to go into a process known as fermentation. So during this time, I'm working out, my muscles are going to start to hurt, right? And it's because they're lacking oxygen. And that force those muscle cells to do fermentation. It's actually called lactic acid fermentation. And so your body takes some of the products from glycolysis and breaks it down, and that end product is lactic acid. So your muscles are going to produce that lactic acid, and that lactic acid is what's actually causing you that pain when you're working out. It's because you ran out of oxygen. I've noticed everybody's a little bit different, I think. For me, it's generally the day, most of the time 2 days after a really intense workout, that I can feel that buildup of the lactic acid. And like I said, everybody's a little bit different. Everybody's body processes things differently. But it is that lactic acid that's actually causing the pain. So that's why, the reason why I went through that whole example is it's so important to have this vasculature there to be able to bring in the oxygen and those nutrients and to be able to remove those waste products to keep us going. Okay, so as I mentioned, we're going to be focusing on this lecture, the rest of this lecture, we're going to be talking about skeletal muscle. And that's not at all to say that cardiac and smooth muscle aren't important, but for this chapter, especially this lecture, we are going to be focusing on the structure and the action of our 640 skeletal muscles. So everything mentioned through the rest of this lecture is all going to be referring to skeletal muscle. When we look at our muscles, we see that attachments are going to be necessary for movement. So muscles are going to span our joints and they're going to attach to bones. Muscles attach to bone in at least 2 places. So we have the insertion, which is the attachment to the movable bone, and the origin, which is the attachment to immovable or less movable bone. And hopefully these two words kind of ring a bell a little bit because I did mention them back in chapter 8 lecture as well. To kind of give you an example here though, if we look at our biceps muscle, right? That's going to be that anterior part of our arm here. The muscle of our biceps, and sorry this is a little bit blurry, hopefully it's not as blurry on your actual slides, but the biceps muscle is going to have a long head and it has a short head. You can see the biceps brachii right here, long head and short head. So the short head of that biceps, the origin, again, origin meaning attachment to the immovable or less movable bone. That short head origin is going to be at the apex of this coracoid process of the scapula. And then that long head right here, it's going to originate, or its origin is in the supraglenoid tubercle of the scapula. So think, scapular region for both of those heads as the origin. And as a reminder, origin is immovable or less movable bone. Then for the insertion, if we follow these down, we see that both of those heads are actually going to insert into the radial tuberosity of our radius, right down here, right? And so if you take your arm and say you're going to do a bicep curl, right? You do that biceps curl, what's moving in that process? Is your shoulder region moving or is your forearm moving? Your forearm's moving, right? So that makes sense because the insertion of our biceps is at the radial tuberosity right down here, right? So that makes sense that our forearm is going to be what's actually moving because the insertion is the attachment to the movable bone. Whereas our scapula is not moving in that process, right? I mean, if you're moving your scapula to do that, you're definitely not doing a bicep curl the way that you should. You're not going to be training that muscle how you should. So that's why the origin is up here near the scapula of both of those heads of that biceps brachii muscle, right? So hopefully that helps you to kind of remember origin and insertion. Insertion is the movable bone. Origin is immovable or less movable bone. So one other thing I want to mention on this slide is that attachments can be direct or they can be indirect. Direct attachments are also called fleshy attachments. So with these fleshy attachments, they're going to be direct. And you can see an example right here of a direct attachment. So the epimysium, which we're going to look at in a little bit, this is going to be the outer fibrous tissue surrounding a muscle tissue, and it's going to be fused to the periosteum of the bone or the perichondrium of the cartilage. So it's going to be a direct fusion to that bone or the cartilage. That's a direct or fleshy attachment. So for example, in this image, what we're seeing is the external intercostal muscles in between the ribs are direct attachments, right? Because they're attaching directly to that bone, right? These are those muscles attaching to the bone. That's a direct or fleshy attachment. We also have indirect attachments. And with an indirect attachment, we're going to have connective tissue wrappings that extend beyond the muscle as either a rope-like tendon, like in the case of our Achilles tendon, over here in this image. So here you see the Achilles tendon attaching at the gastrocnemius, right down here, that's the insertion, right? Right up here, you can see this tendon right here is also going to be attaching to the calcaneus, that heel bone. Or the indirect attachment can also be something I mentioned a few slides ago, an aponeurosis. So a sheet-like aponeurosis is going to be a thin sheet of connective tissue that actually helps to connect your muscles to your bones. So again, we have our insertion and our origin, two places where we see that muscle attaching, And attachments can be direct or fleshy, where they attach directly to that bone or cartilage, or indirect, where we're going to have either a tendon involved or an aponeurosis. Okay, so now we're going to start kind of breaking down the structure of our skeletal muscle. And as a reminder, skeletal muscle is going to be an organ. So it's made up of different tissues, and it's going to have three features. It's going to have the nerve and the blood supply. It's going to have connective tissue sheaths, and it's going to have attachments. Really, if you look at skeletal muscle, it's almost constructed like a really sturdy piece of rope. So here is a breakdown of the different connective tissues of muscles, because the muscles are made up of a lot of different things. So we're going to start big and work our way down. So here at the top, you can see we start off with the muscle at the organ level. So we have its tendon connection, right? This muscle right here is going to be attached by a tendon to the bone. So that tells us right there that's an indirect attachment, right? And we're going to have that fascia that covers it. That's a covering muscle. Then, as we break down into smaller organization of the skeletal muscle as an organ, so this is our skeletal muscle right here, right? This portion right here, it's the actual muscle itself. Then we see that we can break it down into what's known as our fascicle. That's this right here. What's next in the breakdown process, right? So the fascicles are going to involve things like our blood vessels. There's neurons and veins. So you can see all those things in here, right? So this is the fascicle right here. We have our blood vessel in red. We have our nerve right here in the yellow. And this fascicle right here can be broken down further, right? So it's made up of smaller bundles. You see all these small bundles inside that fascicle? Those smaller bundles are termed our muscle fibers, which, if you remember, I told you the muscle cells of skeletal muscle are also known as muscle fibers. So these muscle fibers are technically the cells of your muscle, right? So the muscle fibers are going to be grouped together, and each one is a cell, so it's going to have the typical cellular organelles that we think of, right? So it's going to have a nucleus. You can see the nucleus here in blue. It's going to have the sarcolemma. You can see that sarcolemma right here. That's going to be the plasma membrane of that muscle cell. And the muscle fiber is going to be made up of multiple smaller rod-like structures called myofibrils. So each one of these is going to be a myofibril. And then the myofibrils are going to be further divided into the filaments, also known as your myofilaments. So these are smaller rod-like structures, and we refer to them as thin and thick filaments, as we'll talk about later in this lecture. So as you can see, there are many parts to this muscle, right? You start out at the muscle organ level, goes down into fascicles, the muscle fibers, which are the cells, then the myofibrils, and the thin and the thick filaments. So we're going to kind of talk more about all of these different parts in the following slides. So each muscle, or each skeletal muscle, as well as each muscle fiber, is going to be covered in connective tissue. And the purpose of that connective tissue is going to be to support the cells and to reinforce that whole muscle. They need a lot of structure to keep our muscles from doing things like tearing, and also just to help maintain the structure of the muscle during its function. So here we have the three main parts that I'm going to talk about here in terms of the connective tissue sheaths of skeletal muscles. We have the epimysium, the perimysium, and the endomysium. So first we have the epimysium. You can see it right here, right? That epimysium is the largest part. It's going to be dense, irregular connective tissue that surrounds this entire muscle organ, right, along that whole outside. It can blend with the deep fascia, And you can see here, again, that the epimysium is going to be the outermost part. Then on the inner part here, what you're seeing is that there's all these individual columns. So all these individual fascicles, and those are each going to be surrounded by paramysium. So the paramysium is what wraps each one of these fascicles, and a fascicle is just going to be a group of muscle fibers. So the epimysium is on the outside of this big muscle. Then on the outside of each fascicle is going to be the paramysium. That's that fibrous connective tissue surrounding the fascicles. And then within each of the fascicles, there's going to be a muscle fiber. So you can see this muscle fiber right here. And that muscle fiber is also going to have a connective tissue sheath. So that muscle fiber is encased by another type of connective tissue, which is made up of fine areolar tissue. And this is called the endomysium. And endo means inner, so that's the innermost layer. Epi means outside, like your epidermis, right? So epimysium is on the outside. So if that helps you to kind of remember it, epimysium, paramysium, and then your endomysium. So the endomysium of the muscle fibers are going to connect to each other to form a very loose complex within a fascicle. Small blood vessels and motor neurons are going to pass through this endomysium layer to support and activate each one of these muscle fibers. Over here in this image, what you're seeing is a cross-section of a muscle organ. And you can see how this epimysium is on the outside here, right? It's all the way on the outside. It's that outermost external sheath again. The paramysium is going to be the one that's surrounding each individual fascicle, right? And then the endomysium is going to be the most internal, right? And it's the deepest layer of connective tissue. It's enclosing each one of the individual muscle cells. So again, 3 different types of connective tissue sheaths for our skeletal muscles. The epimysium on the outside, then that paramysium, and on the inside is the endomysium. So I've heard it both ways, mysium as well as mesium, whichever way is better for you to pronounce it. I've heard different people say it different. So this figure is from your e-text. It's table 9.1, and really it's a nice summary of what I just went through on that last slide. So again, that epimysium is going to surround this entire muscle, that organ level, right? Then we have our individual fascicles, and our fascicles are surrounded by this paramysium right here. And when we break down to the individual muscle fiber, the fiber is made up of a bunch of little myofibrils, and the muscle cell is going to be surrounded here by this endomysium. So again, 3 different layers of that connective tissue sheath. Now we're going to go more into the microanatomy of the muscle fiber. And the thing to note about muscle cells is that muscle fibers are very long, right? And they're made up of things known as myofibrils. Another thing to note is that these muscle cells are also going to have multiple nuclei. So you can see in purple here how there's multiple nuclei, and this entire thing right here, this whole muscle fiber, is going to be a cell. So there are also mitochondria in here. This is pointing to one of those mitochondria. Obviously, it's really important for our muscle cells to have a good amount of mitochondria, right? Because we're going to be doing a lot of cellular respiration in these muscle cells. It's also going to have a sarcolemma. So you can see this right here. This is the plasma membrane of your muscle cell, right? Some of the things that you can't see on this picture, but they do exist in your muscle cells, is going to be things like glycosomes and myoglobin. So the glycosomes are going to be located in the muscle fiber to function in things like glycogen storage, because glycogen, again, is going to be stored sugar, and when it gets put into use, it's going to be converted into glucose, which goes through glycolysis to create ATP. We also are going to have myoglobin in our muscle cells, right? And myoglobin is where our oxygen is stored. And of course, you know that with muscle cells, they're going to use up a lot of oxygen, right? So when the oxygen is needed, that myoglobin releases the oxygen and we can undergo the regular cell functions. So again, you can see the different parts of this muscle fiber. We'll talk more in a future slide about the dark A and the light I bands. But just kind of pointing out the other things on here. You have that sarcolemma, which is your plasma membrane. You have mitochondria in here, multinucleate, as well as it's going to be made up of all these myofibrils. So how can you tell that a muscle fiber is a cell? Well, if you look at it, it has organelles, right? And we know that our cells have organelles. So up here is the typical cell that we learned about back in Unit 1. Down here is your muscle cell, right? And a muscle fiber is a muscle cell that has modified organelles in it. So it's going to have things like a nucleus. You can see here, again, it's going to be multinucleate. There's generally multiple nuclei per each muscle fiber. We also have the sarcolemma on the outside here. That's going to be just like this plasma membrane up here on a normal cell. Additionally, we have the sarcoplasmic reticulum. That's being pointed out right here. It's kind of this web-like yellow structure. And then we have these T-tubules in red right here, which we'll talk more about on a future slide as well. But this sarcoplasmic reticulum in yellow is very much like our smooth endoplasmic reticulum in blue up here. So within each muscle fiber, or in other words, each muscle cell, are going to be a lot of rod-like myofibrils that extend in parallel the entire length of the cell. And that's going to give the muscle cell a dark stripe or band and a light stripe or band, which we're going to talk about in a little bit. So that's kind of why you're seeing the different coloration up here. But again, a muscle cell is a cell because it has these different types of modified organelles. So one other thing I wanted to point out, it has sarcoplasm, all this liquid here in red. That is very similar to the cytoplasm up here in a normal cell. So again, a muscle fiber is a cell. It has modified organelles. So looking at a couple of these modified organelles in the muscle cell, I want to talk a little bit more about the sarcoplasmic reticulum and the T-tubules. So the sarcoplasmic reticulum is very similar to the smooth endoplasmic reticulum of regular cells. So the sarcoplasmic reticulum is a network of smooth endoplasmic reticulum tubules that surrounds each one of those myofibrils. There are terminal cisterns that are going to form perpendicular cross channels at the A-I band junction. And don't worry, we'll talk about the A bands versus the I bands in just a little bit. In terms of the main function of this, it's going to be to regulate the intracellular calcium levels. And when we talk more about muscle contraction in our next lecture, you'll see why calcium is so important. The sarcoplasmic reticulum is going to store and release that calcium into the muscles. And this is kind of like an initiator to tell our body when that muscle needs to contract every time that calcium is going to be released. We also have these T-tubules, and this is going to be these are those tubes that are formed by the protrusion of the sarcolemma, which again is that plasma membrane, deep into the cell's interior. This is going to increase the muscle fiber's surface area greatly. It allows electrical nerve transmissions to reach deep into the interior of each muscle fiber. And these tubules are going to penetrate the cell's interior at each A-I band junction, which again, we'll get to that whole A-I band in just a little bit. So here's a picture that hopefully helps take what I talked about on the last slide and kind of visually show you. So within these muscle fibers, we have the sarcolemma. So right here in white, you see that sarcolemma, and again, that's going to be the plasma membrane. There's different tubules. There's going to be the myofibrils and the mitochondrion. But the thing I want to point out right here is in bold right here, and this is this triad. So it's showing you that right here. The triad is going to be the area that's formed at the terminal cistern of one sarcomere. So this is a sarcomere, right? And here's the tubules of that sarcoplasmic reticulum here in blue, right? And the terminal cisterns of that sarcoplasmic reticulum are right here and right here, right? There's two of them. So right here, and that's where we see this triad is formed. So you can see how this is going to be a sarcomere, right? And it's going, pretty much what we see is that the triad is going to join different sarcomeres. So the white tubule, this white tube right here, is going perpendicular to the sarcomeres, right? Perpendicular, sarcomeres are going this way, the white tubule is going this way. And this acts as voltage sensors. So they're getting the response from the brain so that they can change shape in response to an electrical current. So the tubule proteins are going to act as voltage sensors that change shape in response to an electrical current. So this white, I should have mentioned the white tube is your T tubule, right? So the function of those T tubules is going to be to act as the voltage sensors, right? They're getting that response from our brain saying, hey, we need to change shape because there's an electrical current, and that is kind of, we'll see why that's important with muscle contraction when we get to talking about the actual muscle contraction. So we have the sarcoplasmic reticulum, the cistern of them, these blue membranes right here and right here, the terminal cisterns right here, and that T tubule in white, those make up what's known as the triad. And those sarcoplasmic reticulum membranes are going to have integral membrane proteins that actually protrude into the intermembrane space. So sarcoplasmic reticulum integral proteins are going to control the opening of calcium channels in the sarcoplasmic reticulum, like I mentioned on the last slide. And again, you'll see why that's important for muscle contraction in our next lecture. So what happens is when an electrical impulse passes by, the T tubule proteins, here in white, they're going to change shape. That causes our sarcoplasmic reticulum proteins, and those are here in blue, causes them to change shape, and that causes the release of calcium into the cytoplasm. And this is what helps to cause our muscles to contract. Something else you can see in this image are the striations within the muscles. So these are going to be stripes that form a repeating series of dark and light bands along the length of each myofibril. So that's going to be the tubes in this picture, right? Each one of these little tubes is the myofibrils. You can see them pointed out right here. So in terms of the darker regions of the muscle, that's going to be our A band. So you can see how it looks darker right here. Right, the dark red, that's our A band. It's the dark regions. The H zone is going to be the lighter portion in the kind of the middle or middle region of each of these A bands. The M line is going to be this line right here, and that is the line of protein, myosin, that's going to bisect this H zone vertically. Then we have our I bands. The I bands, so right here and right here, what do you notice? They're lighter in red, right? So these are going to be our lighter regions on the outside of each A band. And then we have our Z disc on each of those I bands, kind of looks like it's in the middle of the I band. This is kind of a coin-shaped sheet of proteins that's on the midline of each of the I bands. And we'll talk more about these different regions as we go through the following slides too. So really, myofibrils are going to be modified organelles. They're any of a number of organized or specialized structures within a living cell, and they account for about 80% of the muscle cell volume. So the myofibrils are going to be very densely packed, very elongated, right? And a single muscle fiber can contain thousands of myofibrils. So the features of myofibrils is that they have striations, right? They have sarcomeres, they have the myofilaments, and they have molecular composition of myofilaments. So again, these myofibrils are really important. They're going to make up about 80% of the muscle cell value. Looking more at these myofibrils, probably one of the most unique or distinguishing features of skeletal muscles and these myofibrils are going to be the striations, or those lines. So cardiac muscle is also going to have striations, but remember, cardiac muscle is involuntary. So that's what makes it cardiac muscle. Whereas skeletal muscle is going to have these striations here, but it's voluntary, and we consciously control it. So when we take a piece of that tissue and we slice it and put it under the microscope, we can see these stripes or these striations that are formed by what we label as the dark A bands and the light I bands. So you can see that here, the dark, light, dark, light, dark, light. You can see the alteration of those. So if it helps you to remember, A bands are dark because there is an A in the word dark. I bands are light. There is an I in the word light. So if that helps you to remember it, definitely use it. But with these striations, they're just technically these stripes of the dark A bands and light I bands, and they just alternate over and over again. So when we break this down even further, still looking at our myofibrils, we have what are known as sarcomeres. And sarcomeres are going to be the smallest contractile unit or functional unit of that muscle fiber. So there's a ton of these, right? Because there's so many myofibrils, that means there's even more of these sarcomeres. And they connect together end to end. So they're going to connect, and it's kind of like boxcars on a train, right? How they just connect end on end. So they're lined up within that myofibril, and then they're repeated, right? Over and over again. So the rod-like structures are repeated throughout that actual muscle fiber. And again, there's a ton of these sarcomeres. Again, we can see that we have our A band right here. That A band is going to be dark, right? You can see it's darker here, right? And then we have our light I bands. So you can see how there's a portion of a light I band on each side. So this I band plus this A band and this I band, that equals one sarcomere, right? So the sarcomere, it's a functional unit. And again, it's going to consist of an A band with half of an I band on each end. So from here to here again, just to remind you, that's one sarcomere. The Z disc is going to be a line of these coin-shaped proteins. You can see the Z disc here, the C disc here. It's in the middle of that I band, and it's a line of the coin-shaped proteins, and that's where the thin filaments are actually going to attach, creating what we call that Z disc. So when the sarcomere contracts, what we see happening is that those Z discs are going to come closer together. So this Z disc and this Z disc are going to come closer together during contraction. And one thing to note in some supplemental videos that you may watch, if you decide to, sometimes this Z disc is also referred to as the Z line. So I just wanted to point that out. Synonymous terms there. So basically, this sarcomere is going to be the area between the two Z discs. So going from Z disc to Z disc, sarcomere region there. So you have half a light I band on one end, half a light I band on the other end, and this A band here in the middle. Again, I know I've said that over and over, that's what makes up your sarcomere. And then again, those individual sarcomeres are going to line end to end along the myofibril, just like the boxcars of a train. So you can see one sarcomere from the Z disc to Z disc here. That's one sarcomere. And then it goes straight into another sarcomere. So you can see the start of the I band at the next sarcomere, A band, there'd be another I band there, so just keep going and going along that myofibril. So again, that's your sarcomere. It's the smallest contractile or functional unit of a muscle fiber. So up here, you're seeing an electron microscopy image, and this is a relaxed sarcomere. So you can see this dark A band, right? Going from here to here is that dark A band. You can see the light I bands. Notice that within one sarcomere, there are 2 I bands, 1, 2, and only 1 A band. You can see the Z disc right here, right here. You can see this lighter region in the middle of the A band, which again is going to be that H zone, right? And then you can see that M line as well, right in the middle right here. So the I band is going to be made up of a specific type of contractile proteins. The A band is made up of a specific type, or made up of specific types of proteins as well. And that's what we're going to look at in a few slides. But I just wanted to point out that underneath this electron microscopy image, you can kind of see these different portions being pointed out. So again, light I band, dark A band, there's your Z disc, and this H zone right here. And then what these are showing you in these different colors of red, yellow, and blue are the different types of contractile proteins that we'll be covering still in this lecture. So I mentioned that we have different types of contractile proteins, and that's what I want to talk about next. I've mentioned the I band is the light regions, right? A band is the dark regions. But what makes up those different bands? So the I band right here, as well as over here too, but your I band is going to be made up of actin myofilaments only. So these are going to be thin filaments, which is why they're lighter, and they're going to extend across the I band, and they go partway into your A band. So these actin are going to be shown to you in blue here. So you can see they're all the way in this I band, and then they go partway into your A band as well. So each one of these blue rope type structures, that's showing you the actin thin filaments. They're going to be anchored to the Z disc. So your Z disc is right here, and you can see that anchoring of each one of these actin filaments. That's why if you were to do a cross section and look at the I band, you would only see these thin actin filaments. If we look at the A band right here, you can see that it's not just one type of filament, right? So the A band is going to be, it has both myosin and actin in it. So the thick filaments in red here are going to be myosin. The thin actin filaments we talked about before, they're here in blue, but they're also extending into that A band. So the A band is going to have both actin and myosin in it. So they're going to extend the length of the A band, and they're going to be connected here at the M line. So the M line is where we see this connection between the myosin filaments, right? The actin filaments is going to stop before that M line. It only extends partway into that A band. One more thing that I want to mention here, you notice we have another color, right? We have this yellow color right here. So the yellow is going to be an elastic filament, and it's composed of the protein titin. And it's going to hold the thick filaments in place, and it helps with the recoil after stretch. So this resists excessive stressing so the sarcomere doesn't pull apart. So you can see that titin in yellow is connected to these thick filaments. It's also connected at that Z disc, right? And that's going to help to allow that recoil motion after being stretched to make sure that we don't stretch that sarcomere so much that it pulls apart. So when we go and we look at the sarcomere underneath the microscope, we have a really orderly arrangement of the myofibrils and these filaments, the actin and the myosin. So actin and myosin are contractile proteins, and again, they're going to be very orderly arranged in the sarcomere. So in the cross section, we can see the different types of myofilaments in each part of the sarcomere. So again, this is one sarcomere going from the Z disc to Z disc, right? That's just one sarcomere. We have our M line right here in the middle, right? Then we have it's in the middle of our A band. So from here to here, that's going to be our A band. And our M line, again, is where the thick filaments are going to attach, and they're going to be linked by an accessory protein called myomycin. So the myomycin is going to attach these thick filaments here to this M line, and that's creating the A band. So when you look down here at this H zone, this lighter region right here, we're going to see only these thick filaments, right? We're only going to see the myosin. If we look at the M line, we see these thick filaments that are linked by this accessory protein that I just mentioned called myomycin. So we only see this myomycin right here at the M line. The whole H zone, if we cut at the H zone though, we're only going to see the thick filaments. So again, this is just like if we were looking at this in cross section. Now if you look at the Z disc region over here, if we're looking at the outer edge of the A band, so I guess not quite the Z disc area, right here at the outer edge of the A band, we're going to see that the thick and the thin filaments overlap. So that's where we're going to have both myosin, those thick filaments, and actin, the smaller filaments. So myosin is in red, actin is here in blue. So again, that actin doesn't go all the way through the A band, it stops, right? So it's just a portion of that A band where we have both actin and myosin. So that kind of shows you these different cross sections here of the different regions, and again, different types of contractile proteins. We have myosin being these thick red filaments. We have actin being these thin blue filaments. We have the elastic filaments being titin here in yellow, and again, they're going to attach to the thick myosin filaments to help with that recoiling process. So again, very orderly structure that we see here with these contractile proteins. So looking a little more at myofibrils and the myofilaments. So the myofilaments are going to be the structures that are made from protein, and the contractile myofilaments are going to help the muscles contract. So this is a little bit of what we've already talked about, right? Again, the thin filaments are made up of actin. They're here in blue. The thick filaments in red are made up of myosin. So here we're seeing a small part of one myofibril enlarged to show these myofilaments. So the myofilaments are our actin filaments and our myosin filaments, right? And as a reminder, each sarcomere is going to extend from a Z disc to a Z disc. So this is kind of just showing you again things that we already talked about, but as a reminder, those actin myofilaments, they're the thin ones in blue here. Those are going to extend across the I band, right? That whole I band has that actin myofilaments, and it goes part way into the A band, right? The myosin myofilaments, the thick filaments in red, are going to extend the length of this whole A band, right? And they're going to be connected here at the M line. So next we're going to look a little more first at myosin and then at actin in the next slide. So in this slide we're going to be talking about the thick filaments, which is your myosin. So thick filaments are composed of the protein myosin, and it contains 2 heavy and 4 light polypeptide chains. The heavy chains are going to intertwine to form the myosin tail region. So this, you can see how they're intertwining, kind of forming like a rope, right? This is that tail region of the myosin molecule. Those 4 light polypeptide chains are going to form this globular head region of the myosin. And this head region is going to have an ATP binding site and an actin binding sites as well, which are going to come in to play when we talk about muscle contraction. So during contraction, the heads, these actin heads, are going to link the thin and the thick filaments together. So this links to our actin, right? Because we have the actin binding sites. And this forms what we term a cross bridge. So the heads are going to kind of anchor themselves onto the thick and thin filaments as they move. So the little golf clubs, that's what they kind of look like, right? Sticking off of this flexible hinge region, and then you see that myosin tail portion behind it. So the conformation change really happens right here in this conformation of this hitch, right? We're going to be getting what we term a power stroke, as the myosin heads are going to break that ATP bond of that last phosphate, break it down into ADP, and then they use that energy to move this flexible hinge region and pull that actin just a little bit closer to the M line in the middle. So something that you'll notice is that these myosin heads are kind of offset from each other, if you look at it here, and that's on purpose. So it results in kind of this staggered array of the heads at different points along this thick filament. So that is our molecular composition of myosin. Next, we're going to look at actin. So here we see the molecular composition of our thin actin contractile filaments. And the thin filaments, again, are composed of a fibrous protein called actin. Actin is a polypeptide made up of kidney-shaped G-actin, which is globular in shape, so they're G-actin subunits. And the G-actin subunits are going to bear active sites for that myosin head attachment during contraction. So those active sites are what's going to bind to that myosin head. The G-actin subunits are going to link together, forming a long fibrous F-actin, or a filamentous type of actin. And the 2 F-actin strands are going to twist together to form that thin filament. So you can see that here, your actin subunits. Here you see that little myosin binding region right here. And 2 more things that I want to mention here that are involved in this molecular composition is we have our tropomyosin and troponin. Both of these are going to be regulatory proteins that are bound to the actin to make sure that it binds correctly. So you see this troponin in bright yellow, and then you see this tropomyosin, that's this kind of little thin rope that goes around the actin. And again, when we talk about that actual contraction process in the next lecture, you'll see those come into play. So looking a little more at the molecular composition of myofilaments, we have more than just actin and myosin and tropomyosin and troponin, right? We also are going to have things like nebulin, and nebulin is going to help maintain the alignment of those actin filaments in the sarcomere. We have something else known as dystrophin. Dystrophin is going to link our thin filaments to the proteins of the sarcolemma, and this is going to stabilize that sarcolemma, which again, that's going to be our plasma membrane of our muscle cell. So you can see that kind of in this image, it's helping to kind of link things. Trying to see right here. So this dystrophin is in this teal blue color right here. And you can see how that dystrophin is linking your myofibril to your sarcolemma, which is this light blue line, right? And then it's linked using this membrane protein here in green. So this dystrophin is really important because it helps to transmit that tension generated by the sarcomeres to the tendons. And in terms of the dystrophin, it's going to stabilize and protect our muscle fibers. Again, it's located between the sarcolemma and the outermost layer of myofilaments, and it is the link between the outside and the inside of the muscle cell. So we have the DMD gene. It's going to provide the instructions for making this dystrophin protein. And if we have a mutation in that DMD gene, that's what causes Duchenne and Becker forms of muscular dystrophy. So people with Duchenne muscular dystrophy are going to have a shortage of this dystrophin, this teal structure, in their muscles. So this leads to their muscle fibers being damaged and a gradual weakening of the muscles. Because again, we don't have that important connection between the inside and the outside of the muscle cell. So in muscular dystrophies, abnormal genes or those mutations can lead to muscle degeneration. Most forms are going to begin in childhood. Damaged muscles are going to become progressively weaker. Most people who have this condition eventually need a wheelchair. Other symptoms include trouble breathing, trouble swallowing. I actually had a friend with the Duchenne form of muscular dystrophy. He died when he was about, I think he was 16. He was in a wheelchair by the time he was 12. Very poor quality of life, right? When things like breathing and even eating become a struggle, it's just a very slow progression, but it helps to illustrate the importance of making these proteins and the proteins being formed correctly, right? Because when we have a mutation in that DMD gene, we're not able to make this dystrophin correctly, and that can lead to this Duchenne muscular dystrophy. So when we talk about muscle contraction, and again, we'll be talking about this more in the next lecture as well, but we have what's known as the sliding filament theory in terms of muscle contraction. So contraction is going to be the activation of those cross bridges between the actin and myosin to generate force. So the contractile myofilaments are going to slide across each other, and that end result as they're sliding is going to be the contraction of the muscle. So shortening is going to occur when the tension generated by the cross bridges on the thin filaments exceeds the force opposing the shortening. And the contraction is going to end when those cross bridges become inactive. So this is how our muscles are going to contract and relax. So here on top, we have what we saw earlier, right? This is the relaxed sarcomere. So contraction down here, contraction is going to be the activation of the cross bridges to generate that force and pull the fibers closer together, as you can see in this bottom image, right? We're pulling things closer together. This is a fully contracted sarcomere of a muscle fiber. You don't see that space here in that H zone anymore, right? You see that this Z disc has moved further in, right? So we're pulling things closer together. It actually looks like it's pulling, but what's really happening is that the myosin filaments, they're not attached to the blue actin filaments. So what's happening is that those myosin filaments are going to be grabbing the actin filaments, right? They're forming that cross bridge, and they're literally pulling themselves across. And that's what's causing the muscles to pull and contract. And again, that's when there's going to be calcium released that causes that contraction. We'll get into those details in the steps. We actually go through the steps of muscle contraction in our next lecture. But what we're seeing is really the sliding motion. That's what's happening here, right? We're kind of sliding. These Z discs are moving closer to one another. So look at the distance between them here, distance between them here. They're moving closer to one another. That contraction is going to end when those cross bridges, again, that where we see that myosin and actin binding, right? Remember those myosin heads bind to that actin subunit, right? That forms that cross bridge. When that becomes inactive, meaning they separate, that is when contraction is going to end. We'll go back to this relaxed sarcomere. So just talking a little bit more about this sliding filament model. And just note, this figure right here is actually not in your e-text. It's just a figure that I really like. When we achieve contraction at the level of the muscle, what's really happening is that in every single one of those little sarcomeres, which are lined up in every single one of those little myofibrils all throughout a muscle cell, every muscle cell and the whole muscle organ is going to do the same thing at the same time, which is pretty crazy if you think about it, right? So when we get that activation for contraction, if the actin becomes available, because we have calcium to move that troponin and the tropomyosin out of the way. So we're going to have contact now between the golf clubs, which are those little myosin heads, and the actin. They're ready to hook up together to do a temporary power stroke. And if we give the muscles enough calcium to keep the availability of actin going, to keep that tropomyosin and the troponin off, and we give them enough ATP to continue to do enough power strokes, we can get the sarcomere to full contraction. And that's what's going to happen. So we're going to go from a relaxed state where we have a relatively long sarcomere resting length to a contracted state. And what I want you to notice between the resting stage and the contracted stage is what changes. What bands, what regions of our sarcomere have changed, what regions of the sarcomere have not changed. So you can see we have changed the I band length, right? So the I band is shorter, right? I band, our I band is now shorter when the muscle's contracted. The H zone, this lighter region right here, is going to be smaller as well, right? Our M line has not moved whatsoever. The M line remains unchanged. The A band also stays constant, right? So when we go from relaxing to contracting, you can also see it in these images as well, whatever's easier for you to view. But notice this A band stays constant, right? Our H zone becomes smaller. Our I band is much smaller, right? It's about half. The I band is much smaller than it was. And the Z discs have obviously moved closer to one another as well. And the full length of that sarcomere, right, is now shorter than it was before because it has contracted. So all of this is what we term that sliding filament model of muscle contraction. And this actually won a Nobel Prize for when we were trying to figure out how do muscles actually contract. And like I said before, we will actually go through the steps of muscle contraction in the next lecture. So if all of this isn't quite making sense yet, wait, because when we cover the second part of chapter 9 and actually walk step by step through how muscle contraction occurs, I think it will all kind of come together for you, hopefully. So here, this animation is kind of showing you what we're going to investigate in our POGL activity for this week, as well as what we'll be talking about in our next lecture. So this is showing you that sliding filament theory. This is showing you what happens during contraction. So you can see here the myosin head in red, how it's going to attach to that actin, the smaller subunit each time. It's pulling itself along, right? So the myosin heads, or the globular heads, they're binding to the actin filament, and it's literally pulling itself kind of like a typewriter. It's going ching, 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 and it's pulling itself across. And this is the movement of contraction that we're going to be talking more about in that POGL activity. You can work in groups for that, or you can work alone if you choose. And then we'll also be talking more about this in lecture 9B. Okay, so that is actually all for this first lecture on chapter 9. Some things I want you to be thinking about after listening to today's lecture is, how does the structure of a muscle help it contract? So we talked very detailed about the structure of a skeletal muscle. How does that help that contraction process? What is the sliding filament theory? We talked about it a little bit. What filaments are involved? What's really happening? And we're going to be discussing the sliding filament theory and the steps required for contraction to happen more in depth in lecture 9B. So again, if this doesn't make sense yet, if you're still like, sliding filament, what? Don't worry. We will get to this more in depth, and again, breaking it down step by step in lecture 9B. So that's why I'm actually, this POGL on muscle contraction, I'm not making that due for this week's assignments. I'm making it due for the next week. It's available to you now, but feel free to wait until we have lecture 9B to actually do this POGL activity if you prefer to do that. That is all for this lecture. Mahalo for watching.